These presentations are some modern terms for medical and aromatic plant research.
First, I would like to thank the Scientific Academy for Research and Technology. First, I would like to thank the Scientific Research Academy for Research and Technology for giving me this opportunity to meet you here today. Also, I would like to thank the Republic of China to give me the opportunity to visit Beijing in 2006. Yes, it's okay. F5. Our presentation titled Some Modern Trends for Medicinal and Aromatic Plants Research in Egypt. Here is an ethic conflict between utilization of genetic resources and conservation. Those are two different ways. Our success way must, or our target, conservation of the genetic resources through utilization. And this is what we can call it sustainable development. Let us know our situation. Egypt is a hyperland area. Egypt occupies about 1 million square kilometers. 86% of this area is hyper-arid, and the lake 14 is arid. Egypt early ratified the UNCCD, United Nations Convention to Combat Desertification. Since 2001, the Desert Research Center of Egypt has become the focal point of the UNCCD, and the National Coordination Committee was established. A scientific committee was formed by many of DRC and other national institutions to deal with this problem. Also, we have a national action plan to combat desertification. Here is the challenge that we are faced. We have a lack or limited resources and expanded demands. Let me take you in a short trip through the history. In the early 1927, the idea of establishing Desert Research Institute was initiated by the King Fuad I. April 1949, a royal decree was issued by King Farouk I to declare the establishment of Fuad I Desert Institute. In 13th December 1950, Fuad I Institute was established. And recently, in 1999, a Desert Research Institute has become a real truth by a Republic Declaration. And this slide, this is the Royal Declaration for our institute, and this is the memorial stamp of it in 1960. Our center consists of 4 main sectors: water resources and desert soils, ecology and dry land agriculture, socio-economic studies, and animal poultry production. Also, we have 8 scientific stations and centers: 3 in Sinai, El Maghara in Middle Sinai, Zuaid in North Sinai, Sad in West Sinai, Shalatin in the east-south of Egypt, Mariut, and Siwa Oasis and New Valley. Also, we have Matruh Research Center project. It's a great project in the west coast of Egypt. Introduction. There is some new trends in medicinal aromatic plants research in Egypt. On this presentation, we will present 2 of those ones. We will review the results of this research which were published in periodicals. The first part is the impact of the exposure to different laser types. Also examine the effect of both seed case and exposure duration on growth and yield and chemical constituents of some medicinal plants in Egypt. The second part, the antimicrobial effect of some volatile oils in single or mixture cases against some enteropathogens from Enterobacteriaceae family. The first part, as we said, the effect of exposure to different laser types. of laser in the production of medicinal and aromatic plants can be considering as a modern application all over the world. It may be no more than 10 papers were published dealing with this issue. In case of Egypt, according to the available information, only 3 studies were published. Laser. Since the early stages of the 20th century, one of the most important discoveries that man does is laser. Many applications of laser are well known in many aspects in life. In peace and more, medicine, industry, wars, and recently agriculture. Scientific research dealing with the laser application in agriculture are recent trends. The laser is a device that emits light, electroradiation, through a process called stimulated emission. The term laser is an acronym of light amplification by stimulated emission of radiation. Here's the electromagnetic spectrum. It consists of 3 parts. The first one is invisible, which includes gamma and X-ray. The second one, the visible light. The third one is invisible, it includes the radio waves. The power is shorted from here to here. So we can consider the gamma has the most power to penetrate. So they use it in surgery as a gamma knife. And the least one is the radio waves. The laser in this visible light place. The laser types, we have many types of laser, such gas laser, chemical laser, and eczema laser. The gas laser, many laser types were discovered, such gas laser using many gases. I think it's mainly comes in the localist gases, such helium and argon and krypton and xenon. If this is the other type, the chemical is considered by a chemical reaction, such, this is the type, type for gas laser, another type for gas laser, like helium, silver, like copper, neon, many types of gas. This is the chemical laser. It's powered by a chemical reaction. It has very high power from 700 to 290 nanometer. This has a fluoride, and the deuterium fluoride laser also is 3800. It is another type of chemical laser, but used in surgery. This eczema laser is a chemical reaction between 2 molecules, consists of 2 atoms. One of them is excited electron state. For example, for this, it's the fluoride emitting, and it's noble gas also, and argon fluoride, krypton fluoride, and many other examples. Many laser types were discovered, but in agriculture aspects, the most famous ones are helium neon laser, helium cadmium, and argon laser. This is how the emission of helium neon can be. The first part is exciting the helium atom to transfer from ground stage to upper stage. But this can be done by electric discharge. Then a colloid between helium to neon to transfer the energy from the helium to neon. Then there is a low in the power of it from, this is the orbit which the excited electrons become 3s2. Then the power will be 20.66 electron volt and reduced to be the orbit 2p4. That power now will be 18.7 electron volt. The difference is the emitting of the laser, and then its rapid loss to the ground stage. The laser history in Egypt. Since long time, many laser applications were known in Egypt, such as medicinal and industrial branches, and in soil leveling. In 1994, the National Institute of Laser Enhanced Science was established. It started as a laser center at the Faculty of Science, then it's become a independent institute, belongs to the Faculty of University. The Egyptian experience, little research has been done on laser application in agriculture aspects. Some research has been done on some crops such wheat and corn, and the other as a photosynthesizer insecticide. Too little on medicinal and aromatic plants. only 2 master theses on SAGE in 2005 and on Basil in 2009, and 1 still in preparation on geranium, and 3 papers, one for anise and cumin, the other for fennel and coriander, and the third for acacia parisiana were published in 2009. So it's very recent. The methodology of the Egyptian experience exposure to seeds, it's not me, exposure to seeds to laser light. I know that laser is very recent. The exposure to seeds to laser light on different times and cases. We are using the helium-neon laser, the helium-cadmium, and the argon ions. The time of exposure is different from 1 minute to 20 minutes. The case of the exposure, wet and dry. This is an example for the researchers. The first one is by Wissam Mohammed Sirajuddin in 2005. Her thesis title, Effect of Laser on Growth and Yield on Active Constituents of Sage Plants. This is the methodology. She used the 3 types of laser, helium-neon, cadmium, and argon, on dry or wet, and she used the times 2, 5, and 10 minutes. This is the conclusion which is fine. The argon was better on time 5 minutes, and dry case for exposure for yield, and on wet quality increase the yield quality. The other paper, effect of laser radiation treatment on growth for fennel and coriander. This is a cooperation between 3 big institutions in Egypt, the Desert Cell Center and the Agricultural Cell Center and the Institute of Laser itself. This is the other one on anise and cumin. The methodology was exposure all the seeds to on dry and wet case for helium-neon laser for another time 5, 10, and 20. Here we can find a dramatically increasing on all the yield components. The mode of action, what is the mode of action of laser? The plant growth is controlling by many enzymes and hormones as gibberellic acid and cytokinin. This is for promoting. And on the other side, for the inhibitor, such phenolic compound and abscisic acid. The red light has an important role on gibberellic acid formation. This means that the complex cycle of the gibberellic acid formation is promoted by red light. Which red light? The red light is induced the beta hydroxylase gene, which make a gene express to be formed the gibberellic acid. This expression is inhibited by the far red light. This means that the monochromatic light, which found in laser, is the only way to promote this gene expression. But the polychromatic light, which are found in the sun, inhibit this information. It was in the past 2 opinions to explain the mode of action of laser. The first one is laser maybe enhance the center of gibberellic acid to production in the cell, or enhance the gibberellic acid to do certain actions for cell and the inferior. Recently, we have this paper by Amira Shawi-Sliman. She used the laser to trade acacia parisiana seeds. These seeds have a difficult to germinate because it has a mechanical dormancy, thick seed coat. She exposure the seed on dry case by using helium-neon for times from 1 to 9 minutes. What she found? Here, this is the explanation. The laser beam may perforating the seed coat. This makes more impermeability for solutions and water, and also enhance this. The growth promoter hormones, such gibberellic acid, was increased by increasing the time of exposure and the wavelength frequency. Also, the indole acetic acid growth promoting also increased by 5. On the other side, the growth inhibitors such abscisic acid and phenols were decreased. Then we can count and know how the laser can do. Part 2 of our presentation, the antimicrobial effect of some volatile oil in single or mixture cases will be investigated again in some Enterobacteriaceae family pathogens. The present work was conducted to study the response of some human pathogens belongs to Enterobacteriaceae family, 3 pathogens, to 6 volatile oils in single or mixture application. We make GC-mass analysis for those essential oils, 4 from Apiaceae family, such coriander, Persistenium crispum, Anisum graveolens, and Perennial Anisum, and 2 from the family Lamiaceae, that's Menta vibrata and Menta viridis. We are found 44 components in the GC analysis. The major component of those plants here, oils, the antimicrobial activities of the essential oil were measured by using minimum inhibitory concentration. The selected pathogens from the Enterobacteriaceae, which were 3, Escherichia coli, Salmonella, Enteric, and Fructus vulgaris. The oil exhibits different degrees of antimicrobial activity on the tested pathogens depending on the chemical composition of them. For case of Del, anise, and spearmint, the oil was significantly in single application. On the other hand, the mixture of coriander, peppermint, and parsley was more effective either single or in mixture when compared with the antibiotic sifardine. All those essential oils, we are compared them with the antibiotic sifardine, or which commonly named by psilocybin. Therefore, some essential oils, either in single or in mixture application, could be a source for pharmaceutical materials required for preparation of new therapeutic and antimicrobial agents. This is the GC-mass first here for the mixtures. Here we can find the minimum inhibition effect. This is for the single application. Here we can find there is approximately no growth here, no inhibition for the coriander. This is the highest one for spearmint. Here is the 2 mixtures. This is the first one. Here we can note clearly the inhibition zone. We have chosen the 3 weeks oil to make the first mixture, and the 3 effective ones to make the second one. Here we can find the result. In the single application, many oils have no effect on those pathogens. But here the mixtures all have the effect. Moreover, in the second one, the low level has the best effect. Conclusion: some essential oil, either in single or mixture, may be a new remedy for those pathogens. After we make the kinetics and pharmaceutical tests for them. This is the reference. Finally, I would like to say shukran for all of you. Thank you. Chishu.
Thanks for the Yasser for listening to the discussion. Question, please.
I don't know, but we can test. We can test for this. We can try to test. Yes. Yeah. But I think the volatile oil maybe has a good effect than laser. I think the ultraviolet may be much better in this case. I'll start from the later one. We are use the seeds itself and exposure them to the laser. The effect on health is still under study, but it's normally safe. It's light. And we use very low power. So the effect of them on health, I think it's not have hazard for this. But we must do research to be sure from this. No. We just expose the seeds and cultivate them and take our results, yield, not make any extraction. But I think some of my colleagues now make research on tissue culture. They exposure the tissue itself for laser to know what can do. Also, we are now trying all those on seeds. We are trying now to exposure green tissue, such cuttings, such buds, what it can do. Till now, I told you, just 10 papers, 3 in Egypt. Thank you. Thank you all for your cooperation. Yasser.